Hi, I'm Olivier, and I'm going to be presenting our sequence-sensitive elastic network, which can capture dynamical elements necessary for human microRNA maturation. This is actually an accelerated version of my presentation, so if you find this interesting, I suggest you go check it out. So, microRNAs regulate gene expression, and structural dynamics play a role in their biogenesis. So here you see the canonical microRNA maturation pathway, which starts with a microRNA gene, which is transcribed in the nucleus. Then it generates a primary microRNA transcript, which is recognized and cleaved by the Drosha DGCR8 complex. Then the precursor microRNA is exported, cleaved again by dicer, and then the mature strand is loaded in the Argonaute protein to exert regulatory effects. And today we're going to focus on this first step, and dynamical effects have been shown to play a role in what the microprocessor recognizes as a signal to determine what is a microRNA and what is not a microRNA. For example, here you see this GHG mismatch motif. And so what we're using to study these dynamics function relationships is the ENCOM-DynaSig-ML pipeline. And so we start with a 3D structure of a biomolecule, then we also have experimental measurements of a function of sequence variants. We mutate them in silico. Then we use ENCOM, a coarse-grained normal mode analysis model, to generate dynamical signatures for every variant, which are basically vectors of fluctuation at every position. Here you see them as a vector, and then we feed these vectors in machine learning models and try to predict the experimental outcome. So the data we're using today is high throughput maturation efficiency data of over 29,000 Mir125A variants using purified microprocessor. And so they were measured using barcodes, incubating all the sequences at once with the microprocessor and deducting the maturation efficiency. So one of the reasons why we're using coarse-grained normal mode analysis is because it's very, very fast. It's a fast way to study structural dynamics at the 3D level. For example, for our 29,000 variants, it takes 3 seconds CPU per variant to compute the full dynamical signatures, so around 6 hours for the full data set on a quad-core modern laptop. So here you see the dynamical signatures with the maturation efficiency color-coded. This is just a zoom in on these positions with the standardized dynamical signature values. This is what we're feeding to the machine learning models. And the machine learning model we're using is simple LASSO regression, which is basically just like linear regression, so independent input variables, but we add regularization, so we minimize the sum of coefficients, so that allows feature selection. And it also allows the independency of the input variables, allows to pinpoint important features back to the structure. So from the 29,000 mutants, we constructed a hard benchmark to ensure that there is no sequence redundancy between the training and testing set. So everything you see in red, the mutations that only affect these positions, are the testing set, and everything in blue, the variants that only affect these positions, are the training set. And so when we train the model using both the ENCOM dynamical signature and also the enthalpy of folding, the predicted enthalpy of folding for the variant, we see that they are highly complementary, so their R squared is more than added together, and together they get an R squared of 0.22, which may seem modest, but we have to remember this is a very hard benchmark where the mutations in the testing set were never seen. These positions were never mutated in the training set. So if we do a more classic 80-20 train-test split, then the performance gets really high. Just to mention, a simple neural network can get slightly better than LASSO regression with an R squared of about 0.28. But then the advantage of LASSO is we can map back the coefficients on the structure. And so we see the coefficient for the folding energy is the biggest in absolute value, is negative, so low folding energy is favored, and then we see the dynamical patterns, and one striking pattern is the learning back of this GHG mismatch motif on the backbone. Then we can use this model to predict theoretical maturation efficiencies for completely in silico generated variants. So we did so far 30 million random miR125A sequence variants. And so if we bin them by folding energy, we see that for the low folding energy variants, which are the ones that tend to mature the highest, we get predicted efficiencies that are higher than anything that was measured experimentally. And then we're actually testing some in the lab. So our top candidate has maturation efficiency 2.56, and it still preserves the mature sequence, so it's easier to measure in the lab. And we also have negative candidates which have folding energy lower than wild type, but still the dynamical signature is unfavorable, so they have predicted maturation of 0. And so in conclusion, the 3D structural dynamics are part of the signal that is recognized by the microRNA enzyme for microRNA maturation. It's really fast to use the ENCOM-DynaSig-ML pipeline, 3 seconds per sequence. And we want to test predictions in the lab. We're actually doing it right now and generalize to other microRNA. And so the pipeline is extensively documented and it can be applied to pretty much any biomolecule. You can follow these QR codes if you're interested. And I thank my lab members, and I want to highlight the fact that Natalia and Gabrielle are also presenting here. So if you're interested in our work, go check out their posters and presentations. Thanks.